This has already been discussed that what I am here or what I am presenting here is due to my students, because there are 30 PhD students and then so many MD and MSc students. Actually, it is their work. Of course, I have guided some of them. And then, of course, naturally, I have these so-called distant students, that is, they had purchased my book and therefore they give me all the feedback that is going there are so many papers, all the things I cannot go through today. And these are some of the things that I am going to present today. That will be some of the important aspects. Of course, there are many other things are also there which I am not touching today, but some other time we can touch. And of course, we have some 4 patents on some of the clinical biochemistry equipment. Let us first start with Immunology of Cancer. There are about 50 papers on that, which our group has published. And that, let us say that there is cancer cell, that it will have that particular antigen on that. This antigen can be recognized by T lymphocytes. Of course, the immunological system against cancer, there will be most of the things are due to the T cells, the specificity. Of course, other mechanisms are also there. And then why that there is the cancer cells are not destroyed. And therefore, the one concept is that this particular antigen is masked by certain sialoglycoproteins, and that we have worked a little on that. And see, for example, if we take the cancer, this is the basic technique that I am telling, that there will be then cancer cells can, if you combine, if you keep with chromium, radioactive chromium will be taken up by the cancer cells, of course all living cells, and then that when the cell lysis is there, that will be released, and therefore cancer cell lysis can be identified by the chromium release uptake. That chromium release, and then it's a release. This is well known, this is a standardized technique, that is, I am not going in greater detail. The one important point here I want to tell is that there is some technique called cell electrophoresis. That is, you are familiar with the protein electrophoresis. Instead of proteins, we can put cells. Single cells can be suspended in some liquid, and that if you put under electrical current, cells will be moved, because the cell surface will contain negative charges, and therefore they will move to positive charges, positive pole. And therefore, that depends on the mobility will be depending on the cell surface charge. And these cell surface charges are mainly by the sialic acid content on the cell surface. If you take carcinoma cells, carcinoma cervix cells, this is the mobility in the x-axis, that is microns per second per volt. And on the y-axis, there is the number of cells. Of course, you have to count thousands of cells like that. Then only you will get a statistically relevant population of cells. And in that case, you can see this cancer cervical cells, that this has the mobility. Whereas in the neuraminidase, if you treat the cancer cells with the neuraminidase will remove the N-acetyl neuraminic acid, that is the sialic acid, from the cell surface. Then that electrophoresis will be reduced. 60 minutes, there will be still further reduction. You can see normal cells, normal cervical cells, that the mobility, this is the cancer cervical cells, this is normal cells, that there is drastic difference. And then biochemically also, if you take the N-acetyl neuraminic acid released from cancer cervical cells, this will be. On the other hand, this will be from the normal cells. Therefore, there is a difference between normal cells and cervical cells. That is due to the N-acetyl neuraminic acid content on the cell surface. Now we go for some other, this is the mice model that we take, that is the EL4 lymphoma cells and C57 black mice. This is it is exquisite that one cancer cell, if you inject the animal, that will be developed and then animal will be killed, will be dead within 20 to 30 days. That is the model. And if you take, if you inject the cancer cell and then if you take about 2 weeks after that, there will be lymphocytes from the spleen cells. There will be immune, we call it as immune lymphocyte. And if that is related with, that is incubated with the cancer cells, the target cells, there will be some amount of lysis. That lysis is taken as the chromium release. And then if you take these cells, and that is separated by low specific gravity cells are separated by BSA gradient. Details I am not going, but we can separate certain particular types of cells. Then naturally the activity will be increased. If you take these particular cells and keep it 24 hours in incubation in medium, and then take that, this is what we call as 24-hour pre-incubation, that also will increase the activity of the lymphocytes. And these two techniques are combined together, the pre-incubation plus BSA gradient separation, then we will get drastically increased activity. That is, you are separating the particular cells that are very active against the cancer. The same thing that is in the lymphocytes, the peritoneal cells also, we can take similarly peritoneal cells activity, peritoneal cells treated with carbonyl iron. Carbonyl iron will remove, that carbonyl iron will be taken up by macrophages and then that can be removed. Therefore, what is remaining is pure lymphocytes. The macrophages are removed. Then activity is increased. Therefore, you are separating the active population. And there also, if you take pre-incubation, that there will be more activity. This is a very relevant thing that, say for example, this line represents the tumor size, that you inject the cancer cells here and within about 4 weeks the animal will die, that the tumor is increasing. Whereas if you take the activity of the lymphocytes in these animals, that there will be increase in the activity to a certain extent. Then afterwards there will be drastic reduction of this, the lymphocytic activity. The anti-cancer activity of the lymphocyte will be decreased by after some time. What happens? What is the reason for that? If you take these lymphocytes, this is the sedimentation velocity, and that you can take, this is the most predominant population, but at the same time this is the activity. This line represents the activity. The activity is very less here. The maximum activity, maximum cancer-lytic activity is by this population, which is only a very small percentage of the population. And therefore that you can identify, that you can separately identify. See, for example, we take the activity at the 0 hour, that lymphocytes are taken from the animal as such. You give the pre-incubation for 24 hours, activity is increased. Pre-incubation plus BSA gradient, that is a particular size is selected here, and then you both pre-incubation, BSA separation, and that activity will be highly increased. So this is actually 3,000 times more the active population can be separated. That is 24-hour pre-incubation, that it will reduce the total number of cells to 75. That is, if you put 100 cells, it will become 75 here. And if you take the population, the isolation of low specific activity cells, cell number of cells will be reduced to 18%, but activity will be 500 times or something like that. And the bigger sized cells taken by the BSA gradient separation, that percentage of the cells will be 0.25% of the total cells. But at the same time, this will have 3,000-fold increase in the activity. Now this is the active lymphocytes are taken, and these are active in the in vivo and in vitro. In vitro, I had already told that this will kill the cancer cells. Whereas in vivo also, that if you inject the cancer cells, I told that even one cell that is injected will kill the animal ultimately. But if you inject these particular active lymphocytes there, then the animal will not take a cancer, that is preventive. Even if the the cancer cell is injected and the tumor is growing, to that these specifically active lymphocytes are injected, that there will be a reduction in the tumor size. Therefore, these are the very early work that we have done. Maybe that the lymphokine activated lymphocytes, etc. Now you might have read in the textbooks, etc. These are the previous work on that such time. At that time, lymphokines were not separated, were not identified. And these are the very early stages of those results. Now we go to oncogenic viruses in humans, as directors have told, that it is well known that certain viruses are associated with human cancers. Well known, Epstein-Barr virus for Burkitt lymphoma and nasopharyngeal carcinoma. Human papilloma virus is associated with the uterine cervix carcinoma. Hepatitis B virus with hepatoma. We were wondering with hepatitis HSV, that is herpes simplex virus, whether is there any association with certain cancers. We have taken some oral cancer, 19 cases of serum samples, of course, the normals 650, and anti-HSV antibodies in circulation in 52% these cells, these persons are positive, whereas 71% in oral cancer. That is statistically significant, but not very impressive, because the antibody against a particular virus can be there in any cancer. But that is not so. See, for example, if we take adenovirus, anti-adenovirus in the same cancer, oral cancer, it is only 47%. Therefore, there is something that HSV is associated with this oral cancer. But that we cannot say that it is the root cause or something like that. We need further work on that. But if you take anti-HSV antibody titer in the oral cancer, most of them are coming in this very high titers, 250, 512, and still further like that. Whereas if you take normal cells, normal persons, that these titer values are in the very low values. Therefore, if you take two parameters, that is the percentage positivity as well as the titer values, these two are taken together. That we call as geometric mean titer, which is only a value that the HSV antibody, the oral cancer versus normal, 42 and 96, statistically very significant. And any person, without any statistics, that we can say that this is very well, there is some association. Now, if you take immunofluorescence, that is this cancer cells are taken, over that anti-HSV antibody is added, and then that is made immunofluorescence. Or you can put the immunoperoxidase. Both will give positive tests, positive in many cases. So for example, if you take HSV antigen in oral cancer, the fluorescence 77% and peroxidase 71%. Normals, it will be very low. Next is that whether this viral DNA is integrated or not. This is very well known that viruses will attach to the host cell, and that in the DNA repair this virus is integrated. And whether that is done in these cases, that was the next question. Therefore, this is, we take the DNA from sample, and that denatured single-stranded DNA. To that we will add the DNA probe is and DNA probe is added. That is, in this particular case, HSV probes are added, and then whether that is that probe is binding to that particular gene of interest. Whether there is a positive that the gene is there, then naturally the probe will be adhered there, and therefore we will get a test will be positive. In that case, we can say that the viral gene is incorporated in that. And that is what is happening here, that these the positive samples are there. Some of them are, few are negative. And if you take the hybridization of DNA from cancer specimens with the HSV probes, that you can see HSV, that 56% of course, cancer cervix also, this is HSV2 with the cancer cervix. HSV1 is mainly on oral cancer. And of course, HPV16 does not have any relation. HPV16 is cervical cancer, that is related with cervical cancer. We have done something with HHV6, that is human herpes virus 6 also. At that time, this is about 20-25 years back. At that time, HHV6 was a new thing. And of course, it was known that Hodgkin's lymphoma and ALL are associated with HHV6. And here also we get 100% positivity as well as the main titer value is very, very high. But very curious thing is that in oral cancer also we have got very high positivity as well as very, very high antibody titer. HHV6 also we have done a lot of work internationally also. This has come with some other foreign countries also we have done, and we have 2 or 3 publications on that. But we have not made much on HHV6. That if you take oral cancer tissue specimens and add monoclonal antibody against that with HHV6 glycoprotein, naturally, again, oral cancer has 100% positivity, whereas normal is 0%. Therefore, there is also something related. The HHV6 is also related there. So therefore, we have done that in the oral cancer. In the circulation, there is a lot of anti-HSV antibodies. In the cell, there is HSV antigen is available or present, and the DNA, the cancer DNA, HSV genes are present. And the oral cancer, HHV6 antibody is present in circulation, and antigen is present on the cell. We have also done in cervical cancer with HPV16. HPV16 is really associated with cervical cancer. I am not going in greater detail because it is already internationally well acclaimed that the cervix is infected with HPV, and then after that viral replication 90% will heal, but very, very small quantity, 0.8% will develop a cancer and invasive cancer, etc. For that, there was one Nobel Prize has been awarded for Zur Hussain, and that he showed that the HPV is integrated into the host DNA in human cervical cancer cells. My work, our work was well above that, about 20, 25, 30 years back, and we have shown that this viral integration in certain cancers. With that, there are so many papers on that, with those virus and cancers. And now we go a small work on Ocimum sanctum. Ocimum sanctum is the tulasi, that is the traditional medicinal plant. There are a lot of work has been already done on Ocimum sanctum regarding that. It is various activities, the antimicrobial activity, etc., etc. But what I am telling that this has not been done previously, and therefore I would like just to say something. There is this work is done in rabbits. The atherosclerosis can be induced in rabbits by feeding cholesterol for a longer period, about 45 days. Then here we are giving Ocimum extract feeding throughout this particular period or after the experimental period. Both ways we have done. You can see that, this is the left side liver, spleen and aorta, and in the normal cases, you can see that in the normal, the green is normal, blue is the control. Control means that you are giving the cholesterol, large quantities feeding in the cholesterol. And then red is that you are giving the Ocimum extract, aqueous extract. You can see that there is the there is drastic reduction in the cholesterol content in all these areas after the aqueous extract. VAS1 treated groups show significant reduction in cholesterol content. This is the aorta sections in those animals. 1 month there is nothing much. 2 months you can see the cholesterol deposits. 3 months, 4 months. Therefore that atheroma is there. Now what happens if you give 25 milligram of the drug? It is not much difference. 50 milligram there is difference. 75 milligram the whole cholesterol is removed. Progressive regression of atheroma along with the increase in the dose of the extract, and complete regression when you give 75 milligram dose. Similarly, normal rabbit liver, normal architecture, and cholesterol-fed animals, that there is fat accumulation and pericellular fibrosis, and 75 aqueous extract, you give complete restoration of normal architecture. Regression of atherosclerosis, that is tissue content of the atherosclerotic rabbits treated with 75 milligram of osimum extract. You can see that this is the original and after the treatment. This is in liver, this is spleen, this is in aorta. Therefore, twofold decrease in aorta and spleen, threefold decrease in liver, that is shown. And there are also many publications on that. Now, one more topic, that is the effect of alcohol on liver and brain. This is the effect of alcohol control 1 month, 3 months and 9 months. That there are hepatocellular necrosis and extensive evacuation observed with the time-dependent exposure of ethanol. That is well known, the previous slide, that is well known in the literature. There is nothing new in that, but these are new things. That there is, the nitric oxide plays an injurious role in the initiation and progression of ethanol-induced liver disease. Control 1 month, 3 months and 9 months. You can see that the nitrite level and nitric oxide for that matter is increasing. Similarly, the transform growth factor beta level, that is also control 1 month, 3 months and 9 months. That you can see that it is a cytokine, and that is described in inducer of apoptosis, and ROS, that is may play an important role in mediating the action of the. Similarly, VEGF, that is vascular endothelial growth factor level, that is also growing on, increasing after 1 month, 3 months and 9 months. And VEGF induces the endothelial proliferation. It is a mitogen for endothelial cells and a regulator of normal and abnormal angiogenesis. Induces vascular hyperpermeability by a direct action of endothelial cells. And that is only a representation. The nitric oxide is responsible for vasodilation, and VEGF will increase the vascular permeability, and that endothelial budding is facilitated by this. And the extracellular matrix remodeling is the basement membrane, and extracellular matrix must be degraded to allow the subsequent endothelial cell migration and proliferation. And this is performed by specialized proteases such as MMPs, etc. This part also we have done quite a lot of work. And then this is the endothelial cells proliferate in response to the growth factors, and VEGF plays a crucial role in the pathological situations. Then as a summary of those particular work, hypoxia is one of the pathogenic mechanisms contributing to liver damage secondary to acute and chronic alcohol consumption. Alcoholism causes induction of several factors including nitric oxide, vascular endothelial growth factor, and transforming growth factor. These findings reflect the existence of an angiogenic process, and new vascular structures are formed to provide oxygen and nutrients. What happens when there is liver cirrhosis already produced? We can produce liver cirrhosis in animals, and can we treat something? Tocopherol acetate showed the best result. We have contributed many things, and tocopherol acetate was a good treatment we found out. Lecithin showed better effect than abstination, and preventive measures were better effective than curative treatment. The antioxidant activity or the inhibition of the generation of free radicals is providing the protection against such hepatic damage. That is also one of the conclusions that we have done on those things. Then non-alcoholic liver disease versus non-alcoholic steatohepatitis. That is, a fatty liver results when the triglyceride formation exceeds its turnover. Therefore, hepatic fat accumulation occurs from a variety of causes, including increased fatty acid delivery to the liver, increased lipogenesis, or impaired VLDL formation. Out of that, we have found out that type 4 collagen and liver fibrosis, there is something special in that. Type 4 collagen hardly exists in human liver, and it increases along with the thickening of the basement membrane in the liver disease. Therefore, an increase in the type 4 collagen reflects the liver damage. For example, acute hepatitis will be that can go for chronic hepatitis, and that may lead to cirrhosis and ultimately to hepatic carcinoma. And this type 4 collagen, you can see that this is NAFLD, that is non-alcoholic, whereas this is alcoholic liver disease. There is statistically very significant difference between these two. The normal is here. And type 4 collagen and disease severity, that is also that you can see that there is the increase. Compared to other markers, serum type 4 collagen may be a sensitive marker for active fibrosis. And the cut value we have found, that is 154 nanogram per ml. And that particular conclusion is that conventional LFT cannot identify non-alcoholic fatty liver disease in patients. Therefore, serum type 4 collagen test can be used as a diagnostic aid for the detection of liver fibrosis before the opportunity for treatment option is lost. A few slides on effect of the alcohol on brain. This particular work has been done by Dr. Sukesh and the group, and there is, of course, I have to significantly reduce the whole work. This is only only a small part of that. Chronic ethanol ingestion in brain, what happens if you take brain weight, and this is the concentration given, that is, the alcohol is given different concentrations. The reduction in brain weight and volume is largely accounted for loss of white matter. Here, the proteins in brain, that also you can see that the proteins are reduced along with the increase in the proteins are reduced along with the concentration of the alcohol is increased. The TBARS is increasing, that is and alterations in enzyme level, AST, ALT is also increasing. The transferases are important enzymes, and they are indications of mitochondrial injury mainly. And the other enzymes, ALP and ESH, in the brain, these are also reduced. Similarly, SOD, that is superoxide dismutase, and GST, glutathione transferase, these are reduced. That is that there is some mainly detoxification process is not taking place. Therefore, alcohol produces many of its damaging effects via the oxidative stress, and the enzymes that is against this oxidative stress, they are reduced. The altered balance of the antioxidant enzymes in liver and brain are caused by the decrease in the key enzymes. There are 45 papers published on the alcohol and liver diseases. Some of them has very high citations. Now I am going for an innovative technology to quantitate the serum proteins. See, there are no single instrument for measuring all the serum proteins for diagnostic significance, and that is a good challenge for the in vitro diagnostic industry. If you take some of these fibrinogen, etc., which are in the milligram quantities, but whereas this can be done by turbidometry. Whereas some other substances, such as ceruloplasmin, etc., having very low quantities in the blood, that will need more sensitive techniques, such as nephelometry. And that light is passing through the sample, and that is passing through 180 degree, that is transmitted light, that detector is there. That is turbidometry. Now, nephelometry, same, more or less same principle. There is differences, but the difference is we take scattered light is taken. The light is scattered here, and 90% detector is put as 90 degree. Of course, it can be many other degrees also. But anyway, this is the major difference between nephelometry and turbidometry. Nephelometry, scattered light is taken, and turbidometry, it is unscattered light is taken. The nephelometry, if you go to it is fine particle, turbidometry, it is coarse particle, whereas here it is laser, ordinary light. Emergence 60 degree in the nephelometry, 180 degree in the turbidometry. But more important is that nephelometry is more sensitive, turbidometry is less sensitive. That is the most important thing. But at the same time, these are both techniques are required for taking the protein estimation in the blood. The turbidometry and nephelometry had been developed decades back, and both were available, techniques were available or instruments were available. Nephelometry was not very popular because of the cost of the instrument. Cost usually about 30 to 40 lakhs, that will. Whereas turbidometry was not very popular due to lack of sensitivity and having. Turbidometry offers high linearity and nephelometry offers greater sensitivity. In other words, turbidometry is used to determine proteins present in high concentrations, while nephelometry is used to determine proteins present in low concentration. Therefore, these were there, but these are different equipments and at a very high cost. Therefore, it will be available if we could combine turbidometry and nephelometry into a single instrument. This innovative technology, this has never been done in the whole world. This innovative technology will have the advantage of both turbidometry and nephelometry. And therefore, that will be a schematic diagram that light source passing through the lens, etc. The sample cell is there. And transmitted light that we take as the turbidometry, 90 degrees, 90 degree, that is the turbidometry. Both instruments can be combined into one instrument, and thereby reducing the cost as well as the efficiency. And such an instrument, that is a serum protein analyzer, a new technique that has been developed, both nephelometry and turbidometry. The advantage is that the instrument will automatically select which is required. If the protein concentration is less, that will automatically select nephelometry, or vice versa also. And moreover, it will also detect the prozone. Prozone, you know that if the antigen concentration is increased, that the antibody concentration is remaining same, that you will get, at first there will be some turbidity that will be increased, and then that will, after some time it will be decreased. That is the prozone. And that prozone effect is very important in clinical laboratory, because if the very large concentration of the antigen of that particular protein is present, then the machine will tell that it is a very low concentration. That is the difficulty with the prozone effect. That can be detected here because the instrument will select 1 second, 2 seconds, 3 seconds, 4 seconds, like that. And therefore, whether it will be initially there will be an increased turbidity, then whether it will be going down, that particular part can be identified by the machine. And then about 25 protein parameters such as cardiac markers, anemia markers, immunoglobulin markers, general proteins, kidney markers, diabetic markers such as HbA1c, microalbuminuria, all these things can be identified by this particular machine. And that is very, a lot of this has been marketed, and that is very easily used in the various laboratories. Therefore, and there are 4 patents are also there. Of course, I have told that there are 30 students plus 2 or 3 students are still there. And one among that, Sugesh Mukherjee, he is present here. Maximum number of papers has been taken by him. He is one of the best students out of these 32 students. Therefore, I congratulate him. With that, I remain. Thank you very much.